Welcome to the Explainer. Today, we're decoding a truly foundational document in the pharmaceutical world, the ICH Q1A R2 guideline. This is essentially the master rulebook for how a pharmaceutical's expiration date is determined, tested, and harmonized all across the globe. But before we get into the nitty-gritty of the roles, we have to look at the problem. Just think about the global supply chain for a second. You might have a medication that's manufactured in a humid, sweltering climate, shipped across a freezing ocean, and then stored in a dry, temperate pharmacy. So how do we actually ensure that a drug's expiration date is accurate and standardized regardless of these wildly different global environments? Well, we need unified testing standards. Okay, let's dive into this. To solve this massive global puzzle, the International Conference on Harmonization, or ICH, stepped in and created this guideline. It specifically defines the stability data package that's required to register a new drug across 3 major regions: the European Union, Japan, and the United States. It's really the ultimate harmonized solution, meaning drug developers don't have to constantly reinvent the wheel for every single country they want to sell their medicine in. So, here's our mental map for today's explainer. We'll start with the global stability blueprint, then move on to testing the drug substance, followed by testing the drug product. After that, we'll hit the environmental testing gauntlet, dive into packaging and water loss, and finally, determining the shelf life. All right, part 1, the global stability blueprint. Look, the world's a huge place with wildly varying weather, right? But regulatory scientists brilliantly simplified this by dividing the globe into 4 climatic zones based on mean kinetic temperatures. Now, this specific guideline addresses climatic zones I and II, which happen to cover the EC, Japan, and the US. By agreeing on the baseline temperatures and humidity for these specific zones, we achieve mutual acceptability, and that just means stability data generated in one region is instantly acceptable in the others. It's honestly an incredible achievement in global scientific cooperation. Moving right along to part 2, testing the drug substance. So, the crucial point here is establishing our shared vocabulary. In regulatory terms, the drug's substance is just the raw, unformulated active molecule. That's it. The drug product, on the other hand, is the final dosage form, the actual tablet in the blister pack, or the liquid in the bottle that's intended for marketing. But right now, we're just talking about that raw substance. And to really understand that raw substance, well, we kind of have to torture it a bit. We call this stress testing. Usually it's done on just a single batch, and we intentionally push this unformulated molecule right to its breaking point. We crank up the temperature in 10-degree increments. We blast it with 75% relative humidity, or even higher. We expose it to light, oxygen, and varied pH levels. Why? To discover its intrinsic stability and map out its likely degradation pathways. We absolutely need to know exactly how and why this molecule falls apart before we can ever hope to stabilize it. Which brings us to part 3, testing the drug product. Now, you can't just test a tiny idealized lab sample and call it a day. Regulators demand hard proof that what you test is exactly what you intend to sell. You need at least 3 primary batches. They have to use the exact formulation proposed for marketing, be sealed in the exact proposed container, and be manufactured to at least a pilot scale, which simulates mass production. This matters immensely because the manufacturing process and the packaging itself can drastically affect how long a drug actually remains stable. All right, on to part 4, the environmental testing gauntlet. The gauntlet kicks off with accelerated testing. We obviously don't always have years to just sit around waiting to see if a drug degrades, so we simulate time using exaggerated storage conditions. We're talking higher heat, higher humidity. This helps predict longer-term chemical effects, and crucially, it accounts for short-term excursions. Think of a delivery truck breaking down in the desert for a day. Accelerated testing proves the drug can actually survive that temporary spike in temperature. Next up is intermediate testing. You can think of this as the scientific backup plan. If your drug fails the intense stress of the accelerated gauntlet, regulators allow you to drop the temperature and humidity down slightly to 30 degrees Celsius and 65% relative humidity. It's a middle ground to evaluate stability if the most extreme conditions just prove too harsh for the formulation. But ultimately, there's absolutely no substitute for the real thing. Long-term testing involves storing the drug at its recommended storage condition for the entire proposed shelf life. This is the real-time, undisputed proof required to legally back up that expiration date printed on the final label. Now, what's really interesting about this is how these dense environmental requirements form such a clear standard matrix. For a standard room temperature drug, long-term testing is kept at 25 degrees Celsius and 60% humidity, and you need a minimum of 12 months of data just to submit your application. Meanwhile, your intermediate and accelerated tests run hotter and stickier, but they only require 6 months of data at submission. Of course, not all drugs sit on a standard pharmacy shelf at room temperature. Biologics, vaccines, certain liquids, they require cold storage. So the guideline outlines specific baselines for these exceptions. Refrigerator drugs do their long-term testing at 5 degrees Celsius, while freezer drugs are tested at -20 degrees. And anything colder than that, like those ultra-cold mRNA vaccines we've heard so much about, is strictly evaluated on a case-by-case basis. Let's shift gears to Part 5, packaging and water loss. When you put a water-based or aqueous drug into a semi-permeable container, like certain plastic IV bags or eyedropper bottles, you run the real risk of water evaporating right through the plastic over time. The guideline draws a hard line in the sand here. A 5% loss of water after 3 months under accelerated conditions is the strict threshold for what they call a significant change. Basically, if you lose more than 5% of the water, the concentration of the active drug becomes way too high and the product fails. But testing for water loss in bone-dry environments can actually be pretty tricky, so the guideline offers a very clever mathematical workaround. Manufacturers can test their product at a higher alternative humidity and then use standardized ratios to calculate what the water loss would be at the drier reference humidity. For example, if you measure water loss at 75% relative humidity, you simply multiply that rate by 3.0 to determine what the loss would be at a harsh 25% relative humidity. It's a really elegant mathematical shortcut that regulators fully approve. And finally, Part 6, determining the shelf life. And this brilliantly illustrates exactly what constitutes a failure during the gauntlet. Again, we call it significant change. If the active ingredient's potency drops by 5%, it fails. If toxic degradation products exceed safety limits, it fails. If the pill crumbles, the liquid separates, the pH swings, or the drug just doesn't dissolve properly in the body, it fails. Any one of these triggers proves the medication is no longer safe or effective. Now, what happens when a life-saving drug needs to hit the market before the full 2 or 3 years of real-time, long-term data is actually finished? In these cases, companies have to make stability commitments. They submit the 12 months of data they do have at approval, make a formal, legally binding commitment to continue testing those exact batches post-approval, and update the regulators to firmly establish the final shelf life over time. Granting that final shelf life, it's never just a guess. It is a highly rigorous statistical evaluation. Regulators map the degradation of the drug over time to determine the exact date when the 95% one-sided confidence limit for the mean curve intersects the failure threshold. In plain English, the math literally has to guarantee patient safety on the very last day of that product's proposed life. So, as we wrap up today's explainer, I really want to leave you with this thought. The simple date stamped on your medicine bottle isn't arbitrary at all. It is the hard-won result of a grueling, mathematically precise global gauntlet. So the next time you check an expiration date, consider the immense, invisible scientific rigor that went into proving, beyond a shadow of a doubt, that that exact dose is safe, stable, and ready to heal. Thanks for joining me today, and keep on learning.